Welcome back to this next video in which we are discussing HPVs and cervical cancer. Okay, so in this final video we are now going to discuss the human papillomavirus vaccinations. But before we do that, I have a few comments that I'd just like to make. So the first of my comments is that this up here is not how you spell persistent. Instead, you need to cross out that A and put an E there instead. So that's the first comment I have to make, that I spelt persistent wrong in the previous video and that you need to replace that A with an E, so I'm sorry about that. The second comment I'd like to make is that I'd just like to go back over this grading of cervical neoplasia because I don't think I explained it as well as I wanted to. So I'd just like to explain it again to make sure that all my viewers are completely clear with what this is all about. So this is a grading of how far the stratified squamous epithelium is along the neoplastic process. And the way that you can see how far it is along the neoplastic process is by looking for dysplasia under the microscope, taking a sample of the epithelium and looking for how dysplastic the tissue is. And the way that you can see dysplasia is that the cells will all be different sizes and will have different nuclei, and how severe the dysplasia is corresponds to how far you are along the way to neoplasia, well, along the neoplastic transformation pathway. So, the more further along you are along the neoplastic transformation, the more properties you have of cancerous cells, the more dysplastic the tissue will be, i.e. the more will be the differences in the cell size and the nuclear size, and that's the way these are defined. So I wanted to make it absolutely clear that the definition of these different gradings is histological. You look at the tissue and then how dysplastic it is determines which grading you give it. It is not, for instance, that the definition of SIN 1 is that it's when you've got HPV infection. No, it's defined histologically, it's just that that corresponds to having an HPV infection. So let me just go over this again. So, cervical intraepithelial neoplasia grade 1, also called low-grade squamous intraepithelial lesion, is the lowest grade of dysplasia. So that's very, very mild dysplasia. You'll only have very mild changes in cell size and nuclear size. And this, we think, corresponds to having an HPV infection in the squamous stratified epithelium, because of course the HPV infection as part of its life cycle makes the mid-zone cells over-divide, and that will give rise to the dysplasia, the mild dysplasia. SIN 2, cervical intraepithelial neoplasia grade 2, is then a step up from that. It's more severe dysplasia, which corresponds to being further along the neoplastic process, and we believe this corresponds to when you've had a persistent high-risk HPV infection that has actually now resulted in integration. So you've got permanent E6 and E7 expression that drives more aberrant cell division, which then causes more of these histological markings that we can see, so a higher severity of that. Then SIN 3, we think that that now corresponds to cells that have gone further along the neoplasia process than just having the integrated viral genome and the permanent expression of E6 and E7. We think that in that clone, another cell has got, after some time, another cell has got a mutation that's taken it further along the way towards cancer, and that new clone now dominates. Or maybe you've had more steps in the evolutionary process than just one occur, but you're further along the neoplastic transformation process, you're closer to being cancer, you've got more aberrant cell division, and the cells show it histologically, so that's more severe dysplasia. So that corresponds to being further along the neoplasia process, but you're not yet carcinoma in situ. So remember, carcinoma in situ is where you're at the point of being cancer, but you haven't quite made that leap to being invasive, and then once you have, you're then squamous cell carcinoma, and we've discussed the 4 stages of squamous cell carcinoma, and I think that's probably very clear by now. Right, so that's all I wanted to go over quickly to make sure that that definition of this cervical intraepithelial neoplasia grading, that is clear. I wanted to really emphasize the fact that the definitions of these gradings are histological. You take a sample of the cells, you see how dysplastic does the tissue look, and you grade it on that. It's just that these gradings correspond, we think, to these things. So these aren't the definitions. This is not the definition of SIN1, and this is not the definition of SIN2. It's defined histologically. That's my key message that I wanted to get across here. Okay, excellent. So now let's go on to human papillomavirus vaccinations. So there are quite a lot of these now. I'm going to tell you about two major ones. So the two major ones that I'm going to tell you about are called Cervarix and then Gardasil. So the first piece of information I want to give you about these two vaccinations is which human papillomavirus types do these vaccinations vaccinate you against, because I'm afraid they do not vaccinate you against every single type of HPV known to exist. We've been a little bit more selective. So Cervarix is a vaccination against human papillomavirus type 16 and type 18, which we know makes complete sense. These are the two bad guys. Human papillomavirus 16 is the ringleader of the high-risk HPVs. It is responsible for around 60%, we believe, of squamous cell carcinomas, so it makes absolute sense to be vaccinating against that one. And 18 is the second in command. It's learning from HPV 16, but it's not quite as good as HPV 16. It is also a mucosal high-risk HPV, and it causes, we think, about 10% of squamous cell carcinomas. So these two are really important, so it makes sense to be vaccinating against them, and hopefully that will hugely reduce the prevalence of squamous cell carcinoma. Okay, Gardasil vaccinates you against slightly more. It also vaccinates you against HPV 6 and HPV 11 as well as HPV 16 and 18. Of course, it includes 16 and 18. It would be no use at all if it didn't include 16 and 18. So remember, 6 and 11 are not high-risk human papillomaviruses. They are actually low-risk human papillomaviruses. They will not cause cancer, but they are the major causes of genital warts. The chances are, if you have a genital wart, that it is caused by HPV 6 or HPV 11 infection. So the people who created Gardasil decided that the way that they could outsell Cervarix is by including HPV 6 and HPV 11 in their vaccination, so that we can eradicate genital warts at the same time as reducing squamous cell carcinoma of the cervix. Okay, so those are the HPV types that are included in these vaccines. Let's now talk about how these vaccines actually work. So firstly, what is actually in these vaccines? They are injections that we give intramuscularly, so we inject them into a muscle, but what's actually in them? So they contain something called virus-like particles, or VLP. So let me explain what a virus-like particle is, and you can probably come up with this for yourself. If you wanted to design a vaccine against HPV, it has to be something similar to HPV, but it can't be HPV, because otherwise the HPV you'll just give them an infection. So what would you do? Remember the structure of HPV. It has a capsid, and then it has the genome inside. What if we just got rid of the genome and just had the capsid? Then we could inject that into someone. It wouldn't cause an infection because you need the genome to actually cause an infection, but we could get an immune response launched against the proteins that make up the capsid, and then hopefully that would then protect you from the real virus, because of course the real virus is surrounded by a capsid. Now, virus-like particles aren't quite the capsid, they are just the L1 component of the capsid. You don't have L2 in the virus-like particles. So, virus-like particles, they consist of the 72 pentamers of L1. So remember back to what I told you about HPV's capsid. It contains 72 pentamers of L1, which are called the capsomeres. And remember, in the real capsid, you would also have the minor capsid protein, L2 as well, 12 of those roughly. In the virus-like particles, we're just going to have the surface of the sphere made up of these 72 pentamers of L1. So I think I'll just draw another picture of this. So let's say this is going to be the virus-like particle, and now I'll show some of these capsomeres. So there's one, there's another one, and remember these are all pentamers of the L1 protein, making up the surface of a sphere. And this is what's going to be contained in these vaccines. So in cervix, you'll have virus-like particles for HPV-16's L1 protein and HPV-18's L1 protein, and in Gardasil you'll have both of those, but also virus-like particles of HPV-6 and virus-like particles of HPV-11. Along with that, you'll also have something to cause a massive inflammatory response to make sure that we get an immune response against the virus-like particles that we're injecting in. So we include also an adjuvant. And remember what an adjuvant is from basic immunology. This is a mush containing loads of proteins from pathogens, generally proteins from horrible pathogens like TB. And the whole idea of putting an adjuvant in a vaccination is so that it will trigger an inflammatory response. When you inject this into a tissue, it will lead to massive inflammation because it's full of old proteins from pathogens that our body is very much so used to and therefore reacts to hugely, causes a massive inflammatory response. It recruits the innate immune system very quickly. And having a massive inflammatory response where you have injected in the virus-like particles is going to then promote us getting an adaptive immune response against these. Whereas if you just injected these on their own, it might be the case that you never get the inflammatory response initiated, and if you don't get an inflammatory response, then you don't get all the antigen-presenting cells coming into the area and then taking, phagocytosing the antigens, presenting them, and then moving over to present them to lymphocytes. So it is necessary to get inflammation at a site where you have injected in something that you'd like to have an adaptive immune response initiated against. And the way we do this is with adjuvant, which is just a mush of pathogen proteins, I think generally from TB pathogens, that will trigger a massive inflammatory response when you inject it in. So, virus-like particles along with adjuvant, and you give this by intramuscular injection. So you inject it into the muscle, and we are now vaccinating girls pre-puberty, so 12, 13-year-old girls with these vaccinations. So how then does it work? What type of immune response do you get? Well, you get a humoral adaptive immune response. You get antibodies being produced, which is something that you didn't get produced when you just had an actual HPV infection. Remember when we talked about the immune response to an actual HPV infection, we said you do not get antibodies produced, and this is the massive advantage of vaccination over just having an actual infection. Okay, as far as the immune response goes. So what this is going to trigger then is the humoral adaptive immune response. And I'd just like to do a brief little flow diagram about the humoral adaptive immune response. And this will be a brief flow diagram. I could talk for hours about the humoral adaptive immune response, but I'm not going to. I'm just going to do a brief flow diagram of it. So humoral adaptive immune response. So of course, what's going to happen is we are going to get B cells being activated, which have the ability to produce antibodies that are directed against the L1 proteins. And of course, you'll have different B cells for the different L1 proteins. So for each of the different types of HPV VLPs that you put into your vaccine, you'll have a separate humoral adaptive immune response being initiated. So we'll just talk about one humoral adaptive immune response for one of these. So what you'll get is the activation of B cells. So I'll just draw a little picture here. So B cells are crucial in this. So this picture can represent a B cell. Here's its nucleus. And these B cells that are going to be activated will be ones that have a design of antibody that is capable of binding to the L1 protein. So let's say we vaccinated you with the Gardasil vaccine, and let's follow the humoral adaptive immune response against the HPV6 type. So HPV6 type has a certain L1 protein. That L1 protein will have a certain structure, and there are loads of possible antibodies you could produce that will be capable of binding to it. Let's say this B cell produces a design of antibody that binds to the L1 protein of HPV6. This is now going to be activated as part of the humoral adaptive immune response initiated by the vaccine. I'm not going to go through the activation process. We're going to just acknowledge that it will be activated. And what will then happen is it will divide. So the first thing it will do is it will divide into loads and loads of B cells, and because I'm lazy, I'm just drawing these as small cells now. Obviously it's a bit out of scale, they should be the same size as this cell, and I'm not drawing their nuclei, so this is just representing lots and lots of these B cells. So this B cell will divide and it will produce loads of B cells that are identical to it, and which therefore all have an antibody design identical to it. Okay, so all of these B cells that we've now got here, and they look strange without nuclei, so I'm going to just put a few red dots in to show their nuclei here. So all of these B cells that we're producing here will now all be capable of producing this antibody that can bind to the L1 protein of HPV6. Now what will happen is they'll start to differentiate. So the next thing that happens is they start to differentiate into plasma cells. So let's do the plasma cells in blue. And the plasma cells have a more oval shape than a round shape. So you'll get lots of these differentiating into plasma cells, which I'm showing here in blue. So these are plasma cells, and plasma cells are special differentiated B cells. So this is a differentiation process that has occurred here, and they are specialized to produce the antibody design that the B cells had, but at a fantastic great rate. So this, let's say this plasma cell came from this B cell, it will have the same antibody design, but it will produce huge amounts of the antibody now. And plasma cells generally, they go to the bone marrow, so I'll draw a little picture here. So if we have a bone drawn here, one of the long bones, so this looks like the femur, so I'll actually turn it properly into the femur, like so. So plasma cells go to the bone marrow, and the bone marrow, of course, is in the centre of the bone here. So they go to the bone marrow, and then in the bone marrow they'll start secreting huge amounts of antibodies, and these antibodies will be going into the blood. So they go to the bone marrow and they start production, and they absolutely chuck out antibodies. So they really do set up production. So now from the bone marrow you'll be getting a huge amount of antibodies being produced, and these antibodies will go into the bloodstream. So this is what you're going to start activating against the HPV6 L1 protein. So just to summarise again, you'll activate B cells that produce an antibody design which can bind to the HPV6 L1 protein, and of course concurrently you'll have immune responses exactly the same, but different B cells being initiated against HPV 11, 16 and 18 if we're using the Gardasil vaccine. When the B cell has been activated, it divides and produces loads of identical B cells that importantly all have the same antibody design. Then they start differentiating, and a lot of them become plasma cells, which are the production versions of B cells. They start chucking out antibodies, and they go and hang out in the bone marrow, and from the bone marrow they chuck antibodies into the bloodstream, and these are going to be able to bind to L1 proteins of HPV6. Now, a lot of the plasma cells that you produce in a humoral adaptive immune response do not live that long. They might live a month maybe, and then they'll die and be removed. However, some plasma cells live for an incredibly long time and can produce antibodies for years and years and years, going on decades after the initial vaccination. These are called long-lived plasma cells. And these are the ones that are really important for giving us long-term protection against HPV infection. So you get some long-lived plasma cells that will remain in the bone marrow for years and years and decades and be producing antibodies which will be secreted into the bloodstream for all of that time. And these antibodies in the bloodstream, these are what are going to protect you from being able to get an HPV6 infection in the future. And there's a fancy term for this. We call this serological memory. So the vaccination gives us serological memory. It means that we're going to have antibodies circulating in the bloodstream, i.e. in the serum, that are directed against the L1 protein of the HPV6, and they'll always be there protecting us from HPV6 infection. Now, just before I come on to how those antibodies are going to stop you getting HPV6 infection, let me just talk about the fact that not all of these B cells here will differentiate into plasma cells. Some of them will become memory B cells. So some of these will turn into memory B cells. few in comparison to the amount that turn into plasma cells. So most of them turn into plasma cells, but some of them turn into memory B cells. And these are effectively doing exactly the same job as the original B cells. They are capable of being activated if ever you get infected by this thing again. So if ever you get infected by HPV6 again, and the humoral adaptive immune response is initiated again, these perform the same role that this initial B cell did, because it wouldn't do for this initial B cell with the design that we actually need here that can bind to L1 to divide and divide and divide, and for all of these cells to become plasma cells, because the plasma cells can't then be activated to make more. Okay, you need some to replace this original B cell. It's just like when we were discussing the way the squamous stratified epithelium works. When the basal cell divides and produces two new cells, one of them has to remain a basal cell and one goes up so that you maintain the population of basal cells. That's what memory B cells are for. They replace the original cell that you used up in this division process. And of course you end up with more of these than you had originally, and therefore it's quicker to activate this same humoral adaptive immune response the second time than it was the first time, because you've got more of these B cells with this design, this specific design of antibody. And this part of the memory for the humoral adaptive immune response, this is called reactive memory. And the reason it's called that is because it's reactive. You know, if you were exposed to the antigen again, the L1 protein of HPV6 in this case, you would get another humoral adaptive immune response. You'd get another reaction, but this time it would be quicker and it would be bigger because of the fact that you have more of these ready memory B cells here that can be activated by the antigen, and they'll all divide and a lot of them will differentiate into plasma cells. So these memory B cells are replacing the original B cell, and because you've got more, that means that if you're infected a second time by HPV6 and you want to launch a humoral adaptive immune response again against the L1 protein of HPV6, you'll get the response being initiated quicker and it will be bigger. You'll get loads of plasma cells being produced very quickly. Okay, so that's just a little reminder of the humoral adaptive immune response then. Coming back now to the serological memory, because this is the really important bit. This is important just because I wanted to finish the humoral adaptive immune response pathway, but this is what we believe actually protects you, the serological memory. The fact that after having this Gardasil vaccine, you will have, or the girl who has been vaccinated against it, will have antibodies circulating in her blood against the L1 proteins of all of these HPV types. And this will occur for years and years and years and years, decades even, because of the long-lived plasma cells, and this is called serological memory. Now, why does this protect her? Well, this comes back to what I said about the virus entry, and remember I didn't say much about virus entry, so I'm just going to draw a little picture of this. So let's have a basal cell, here in yellow, of our squamous stratified epithelium, and then let's draw the virus, the HPV virus here. So there's its capsid, and of course it's completely out of scale. The virus is tiny compared to the cell. So remember when I talked about how it enters the cell? What I did say, the one thing I said, is that it takes absolutely ages. It is a very slow entry mechanism. It takes absolutely ages. This virus particle will be sitting outside the cell for ages before it actually goes in. Hours, okay? Now, if you've got antibodies circulating in your bloodstream, antibodies also circulate in the interstitial fluid. They go into all the tissues of your body, okay? The idea is that if you've got antibodies in the fluid nearby this squamous stratified epithelium, they will bind to the L1 proteins of the HPV virus particle here when it's trying to enter the cell. And then once you've got antibodies all over this, it blocks the entry mechanism. So if I just draw antibodies all over this, the idea is that having the antibodies physically all over the virus particle stops the virus from being able to get in. So it's not that the antibodies lead to the destruction of the virus particle. It's simply that them binding all over the virus particle stops the entry mechanism, and this is why it's important. It's a really slow entry mechanism, so that the antibodies in the interstitial fluid that have come in from the blood have time to actually bind to the virus. Okay, so overall that is why we believe HPV vaccination protects you against HPV infection. And there is obviously discussion about whether we should be vaccinating boys against these as well, because of the fact that they don't just cause, well, we think they don't just cause cervical cancer, talking now specifically about the high-risk ones, 16 and 18, but we also think that they can cause cancer of the preputial mucosa in boys, so penile cancer, also of the urethral lining, and also of the anus as well. And of course, 6 and 11 most definitely cause genital warts in boys as well. So if we vaccinated boys against those, we'd be able to stop genital warts in boys as well. So there is a lot of talk about boys being vaccinated with these at the moment as well. But at the moment in the UK, I think it's just routinely given to girls between the ages of 12 and 13. So just to summarise this important mechanism that I've told you about here, and then we'll finish the video. The vaccination is a virus-like particle vaccination where you've got these structures that are just 360 L1 proteins joined together, and you launch a humoral adaptive immune response against the L1 proteins of the different HPV types. And here's a reminder of how the humoral adaptive immune response works. You activate B cells which have an antibody design that will be able to bind to the antigen that you're launching the humoral adaptive immune response against. When they are activated, they divide and divide and divide to produce a whole population of identical cells with all identical designs of antibodies, and these start to differentiate. Most of them go into the plasma cells, which are the actual effector B cells, and indeed that's another name for plasma cells, effector B cells. And these are the ones that actually start chucking out antibodies into the blood. They go to the bone marrow and they set up production there. They chuck antibodies into the blood. And some of the plasma cells will be long-lived and will last for years and years and years, and will continue chucking antibodies into the blood so that you've got long-term serological memory. And this is believed to be why these vaccinations are effective in stopping you getting an HPV infection, because when the HPV particle comes along and is about to infect the basal cell, it takes so long to get in because the entry mechanism is so slow that the antibodies have time to bind all over it, and antibodies being bound all over it just stop it from being able to complete its entry mechanism, so they block the basal cell ever becoming infected with the HPV. That's how it produces protection against the HPV, and this is something that the normal immune response against an HPV infection fails to produce. That's why the vaccine is so much better. Okay, and then just to complete our discussion of the humoral adaptive immune response, you also produce loads of memory B cells from these differentiating cells here, which will replace this original B cell, and they can then be activated again if you were presented with the antigen again, and they'll go through this exact same process. They'll divide and divide and divide and then differentiate to produce plasma cells and then more memory B cells. So it's just to replace the original cell that was used up in the humoral adaptive immune response process. Okay, so that concludes this video on human papillomaviruses and cervical cancer. I'm glad you made it through to the final part of the video, and I hope you've learned a huge amount.